and then we could do a quick, address, and then they could, get started. All right. Welcome everyone to today's session to explore agentic AI in our works and to also dream beyond our immediate environments, our rules, and our tasks. Right. This is a really great capability, and we are all thinking about how to really use this to improve our lives and also improve our work and basically our societies and communities in general. Um, we have with us Finn. Finn is in McGill. We'll be going to the Arc Institute in a few weeks. Um, I feel like like the how do you call it? So there is now a race, towards, the golden boot, Mbappe and Messi. Who do you think is going to win? Who do you
Can you?
think is going to win the golden boot?
Yeah, the Golden Boot. That's a good question. I don't know. I'm not loving Messi right now, if I'm being honest. I think the Egyptian game was stolen, and but yeah, I'm okay if that's in the recording. But I think Holland. I want Holland to win.
You're in
I
Holland,
think
okay.
I
Okay.
think France is like they've had too much glory, and we need an up and coming team to win.
So I feel like Finn and I were the, Mbappe and Haaland in the AI in biology, conference. We're trying to see who gets most questions, out to the presenters.
Oh, okay, I see.
And then I feel like every session Finn had a question, which was, really, great. He's definitely, very thoughtful about, the work, that we do. And I think, it's a really great attribute and also one of the important, reasons why we invited him to lead, today's, session. He thinks about the work actually, which is, great, as well. Before he, takes, the mic, a couple of announcements. So Build with Claude is still ongoing. I know like majority of you were not accepted into the the competition, but I think it's just great to try your hands on like the three research projects that they highlighted. It's a great exercise to really see um how to use the tool and how to also use it to solve problems as well. So that's the short announcement I have for everyone. So with that said, Finn, take over. Gonna- Yeah, the floor
Did I
is
just
there. Yeah.
did I just cut up for a second? Okay, yeah, I just had to switch my Wi-Fi because it was messing up. But yeah,
Yeah.
excited to get started. Okay, I'll share my screen. And just to confirm, Samuel, how I have like an hour or 40 minutes and then like tw 20 minutes of questions, something like that. Yeah. Okay, sounds good. Yeah, so today I'm going to be talking about black box and white box data in the age of AI and biology. And I think this is a really important topic because, we're going to be talking, I guess the theme of this lecture series is about agents, but I think one of the most important things that people have to be thinking about is what you put into agents because, like we talk about, agents can do a lot, like we were talking about before. But I think ultimately what's going to determine your impact as a bioinformatician a lot of the time is one, how you think about the problem, but also what you put into these agentic kind of architectures. So I'm talking about this today because, yeah, I think it's a really important topic that everyone should be thinking about. I usually like to start my presentations with some sort of art. So this is a picture, like a painting by Georgia O'Keeffe, who's an artist I really admire, and the shape of this bend uh, is similar. It kind of resembles a structure I'll be talking about today in the brain. But yeah, today I think I'm going to be going over just my journey with research thus far and talking about how kind of each step of this journey I think can inform what bioinformaticians do in the age of AI and biology. So yeah, excited to get started. Um, so yeah, I'll be opening talking about my kind of brief intro of my journey with research. I'll be talking about kind of the importance of theory and white box data and ML research and like in biology. And I'll be talking about an example of on how machine interpretable black box data can accelerate science. And I'll explain what that all is as we go. And then I guess we're gonna conclude with a choice of what you want to hear about because I can talk forever, as Samuel probably knows. So I actually want to hear what you guys want to talk about. I prepared a couple slides, so we'll have a vote around then. It'll be fun. Um, and if we have more time, I can talk about it all. Yeah, so just to start off, I'm from a mystical town called Toronto, Canada. That's where I grew up. If you guys have heard of that, we have tall skyscrapers like the CN Tower. We have great movies. We have great sports teams like the Toronto Raptors. And I've followed basketball, but we recently got Kawhi Leonard, so that's pretty exciting for us. Um, I did my undergrad at McGill, working at an institute called the Neuro, which is like a founding institute of modern neuroscience. It's my amazing lab. This is my friend. This is him in 2 BCE, which I call before Claude era. So it was very, it was an interesting time. We were in the cut trying to figure out all these softwares, and it was fun, but it was definitely slower. Recently, I've started working at the Arc Institute. So I won, like with my two best friends, we applied for this program that they ran where they were recruiting teams across North America to like pitch projects. And we pitched an interesting project and we got selected. And I'll be talking about that project today. Yeah, so that's kind of what I'm doing right now, working remotely for the Arc Institute. And you guys might be asking, like, how did I get here? This is the AN Biology conference that me and Samuel met at. So this is kind of some pictures I found of me and some other people that were talking. I think me and Samuel both after each lecture would go up to the lecturers and talk to them. So we often found I think ourselves next to each other. And we got to talking one time, and I mentioned that I actually have some friends from Ghana that live in Accra, and I think that intermixed with our constant discussion, my lot of questions, I think gave Samuel the idea to maybe ask me to talk. So yeah, thank you, and Biology CSHL conference, and again, thanks for the invite. So yeah. Okay, wait, my slides. So yeah, today, like the first thing I want to talk about is an example of the importance of theory and white box data and ML research. So white box data, I'll just start with explaining that, is the idea that what you're feeding into a model is something that you can understand. It's like something like a gene, like in single cell data, you have your cells and you have your gene count. So how much gene each cell has. So you kind of can understand there's like an interpretable feature to what you're looking at. Um, uh, and that's what white box data is. It's a data type that you can understand and has like real meaning. And I'm talking about white box data and how it relates to the importance of theory because I think what I hear about in this kind of AI and biology world, sometimes it feels a little misguided, and I want to talk about my thoughts on why that is. But just to preface, I don't know, have you guys talked about virtual cell ever, like that kind of paradigm or no? So basically, um, there's this movement going on in AI biology where like computer scientists are trying to build models that can predict how cells respond to certain drug additions. And they basically take single cell data and they're trying to build these big machine learning models of how when I give a certain drug, like drug X, how does it then respond? So yeah, it's called a virtual cell, and I think it's a cool idea. But I think that you really have to think about like what the purpose of a virtual cell is. Is it to predict the effect of a cell to a drug, or is it to enable like new health research? And from my view, I think it's to enable more health research. That's the ultimate goal. And I think a lot of computer science building these virtual cells, it's really cool stuff, but I think there's an absence and a lack of appreciation for theory a lot of the time because I think oftentimes the most important question is not how do we get a cell to respond X way? it's what do we want to respond in what way? You know what I mean? You like people are trying to potentially like in Alzheimer's, they're trying to reverse microglia activation. And people are like, oh, we're going to make drugs to reverse microglia activation in the brain. But with a virtual sound, this is like this big kind of like, look what we can do. But, sometimes I think I'm like, is that actually what you want to do? Like, I haven't seen that much solid evidence that that would actually improve the symptoms of people with Alzheimer's, because sometimes microglia activation would be good. So I want to talk about this topic today just because I think more of what these big models need to be doing is harnessing themselves in real biological questions. And yeah, so this is kind of also how I started my research career.
I have a quick question.
So
I have a
yeah, yeah,
quick
go ahead.
question. So it's the virtual cell, kind of like, is it similar to like a perturbation, study where
Yeah.
you knock out like, gene or like, like a group of genes and see its, effect?
Mhm.
Is it more like, does it happen on like a single cell? Oh, like it's a bunch of cells grouped together, and more like a more like a
Like
massively
pseudo.
parallel approach.
Yeah, I mean, I think it's every the thing that I think you'll find with a lot of this field is everyone has the same word for many different things. So, and no one agrees on like what the words we should use are. So it's like that's honestly one of the most difficult parts about navigating this field, I feel like. And so, I always get like I don't even know, like I don't have a perfect answer, but I'd say that yeah, it's like perturbation response prediction basically. And some people do that on a per cell level, some people do that on a more like sample level. But it's, one example would be gene perturbation, so like CRISPR knockouts. Another would be, what if I give you some sort of drug, like I can't think of a drug right now, but how could that affect, the cell state that
Like
exists?
a
So it's
MEK
just
inhibitor, right? Which really targets,
exactly. So
like,
how would that change what the transcriptome of cells look like, the RNA profile?
okay, okay, thanks.
Um, Yeah. But so I'm not really talking about that today, but I just brought it up because I think it's really a big discussion in the AI biology field, and I think more people need to be anchoring what they're doing in theory, so it can have impact, not just trying to build models. You know what I mean? So yeah, and I'll talk about the reverse of that, how the reverse of that is also helpful. But yeah, this is my intro to research and kind of how I think about using theory and an example of like my story. So yeah, so have you guys heard of multiple sclerosis ever? It's third most common neurodegenerative disease. So it goes Alzheimer's, Parkinson's, multiple sclerosis. Basically, multiple sclerosis is a disease that's typically characterized by the formation of lesions around the brain. So essentially, parts of your brain go missing and you get like these holes. Um, because it's an autoimmune disease, so your immune cells eat away at the brain. But recently, people have recognized that there's another type of damage near parts of your brain that are in contact with cerebrospinal fluid. So if this is the cerebrospinal fluid and this is the border with your brain, there's like a lot of damage close to the border and less as you go away. So anywhere touching this cerebrospinal fluid, so like in the ventricles or surrounding the brain, has something called a gradient of damage. This kind of gradient of damage that can exist at the borders. You can kind of think of it as shading it in with a pencil crayon. Um, and I can skip ahead to so you can like visualize it. But yeah, this is kind of what the gradient looks like. So this is the cerebrospinal fluid. This is the gradient of damage. Um, but yeah, what's interesting is kind of this gradient phenotype is really clinically meaningful. So people have recently found that it's independently predictive of the entire course of multiple sclerosis. And also this kind of gradient of phenotype suggests that something might be diffusing into the brain from the cerebrospinal fluid, allowing the damage to occur. But despite this, and if that were the case, you'd expect something at the barrier to be changing first, kind of to allow this diffusion into the brain. And despite there being some preliminary hypotheses about this phenotype, and despite there being clinically despite it being clinically meaningful, no one had investigated the border between the brain and the cerebrospinal fluid yet. So that's what my team did. This is like one of the first papers I was ever on. So Basically, we took postmortem brains from people that died with multiple sclerosis and postmortem brains from people that didn't have multiple sclerosis. Then we did single nucleus multiomic sequencing. So we collected attack data, single nucleus attack data, and transcriptomic data from individual cells in one cell. So for every cell that we get from here, we have its epigenetics and we have its RNA in the same cell. And basically, my job was to use this data guided by kind of a theory that my lab had about the border, to ask three questions. One, how might the border cells be changing in disease, allowing damage to occur? Two, what's the mechanism that might be causing that to happen? And three, what's turning on that mechanism? And I'm going to be talking about that today as an example. So first, the first question was how do the border cells change in disease? That might they be opening up, allowing diffusion of cerebrospinal fluid into the brain? So basically, using gene expression analysis and differential expression, I found I like kind of these are the cells at the border. So red cells are multiple sclerosis cells, blue cells are non-multiple sclerosis cells. And you can see initially that there's a difference in the cell types. Then when you use differential expression, so like t-tests basically, on what genes are changing between multiple sclerosis and the healthy cells, you find cell junctions are changing.
So,
Um,
Finn, if I may,
yeah.
ask, because I'm not really sure, like, like how many people have done
Oh, single
this
cell. Okay.
before.
Sounds good.
Right? So, maybe like the first question would be,
Mhm.
why, like, why are we using UMAP?
Okay,
Instead
yeah.
of maybe, just like a correlation, plot between maybe two variables or something. And what would this, tell us, differently from maybe other methods, right? I'm sure
Yeah,
people might be interested in this as well.
totally. Yeah. So, I think so. Do people have a background, like have heard of single cell data before, do you think? Or should I maybe
I think maybe give like a little bit, background
have an
so that
intro? Okay, yeah.
we're
Sounds good.
yeah.
So like your whole body's made of cells. You have skin cells, you have teeth cells. And I think something not if you're not in biology, like a lot of people don't think about is that, they have the same DNA. Why is my skin cell different than my teeth cell? You know? It's because both these cells use different parts of the DNA. So my skin cell uses skin RNA and my teeth cell uses teeth RNA. They use different copies of the DNA. So you can kind of just think of RNA as like a proxy or a kind of a representation of what the cell cares about. So again, if I measured my skin RNA, I could see that my skin cell cares about skin. If I measured same thing with my tooth cell. So again, RNA is a proxy or a kind of a representation of what the cell cares about. And if you measure it, that tells you something. So using the same logic, a lot of the time, what people do in like brain research is they take people's brains that died of a disease and said, I want you to use my brain to cure the disease that killed me. We basically take a part of their brain with their consent after they've died, and we kind of sequence the RNA in their cells, just like you could sequence the RNA in your skin cells. We sequence the RNA of the brain cells, and we sequence it from people that died with a specific disease and people that didn't die with a specific disease. And using the same skin tooth kind of paradigm, we ask for a given cell that we find in the brain, in a diseased person, how does the RNA differ from a person without the disease? So by asking a question like that, we can see how are the cells changing in disease, and that's allowing kind of the disease to progress. So again, just looking at how RNA is changing between two states. And here what we're doing is we're asking the question, how is RNA changing between the border cells, like specific border cells in the brain, um, from in someone that has multiple sclerosis and how someone doesn't have, and someone that doesn't have multiple sclerosis. So this is just a visualization of RNA. And what we're doing is we're looking at how the RNA is changing, and you can see the red cells are the brains come from brains of people that had the disease, and the blue cells come from from people's brains that didn't have the disease. And this is just a way to show that the cells are different. So we see that the cells are different between the disease and the healthy people. Um, when we look at why they're different, you can run something called a gene ontology analysis, which basically looks kind of groups RNA genes into like functional groups, so something you can understand. Basically, what you can see is that the junctions between cells are changing between healthy brains and multiple sclerosis brains in the border cells. So that's important because junctions form the connections between cells. So you can imagine if the junctions between cells were changing, that might make the border between the cerebrospinal fluid and the brain more loose, allowing more things to go through. So yeah, the junctions are changing, which kind of gives us an idea of how to answer our first question. How are the cells changing between health and disease? The junctions are changing. So that's kind of the first thing. Then, to answer our second question, what's the mechanism that's causing these border cells to change? I ran a machine learning algorithm that basically asks a question. It says, there's like transcription factors in cells, right? So what you can do is you can correlate how much, when I see this transcription factor, what other genes come up? And by doing that, you can build kind of groups of transcription factors associated with their genes. And then what you can do, because you have this information about like the epigenetics of cell and the RNA, you can then check in the epigenome if this transcription factor that you found to be associated with RNA has places to bind to regulate the genes you're seeing. But, that's like you can, I'm sure you guys can like read up on that if you want, but the main message is that we found kind of robust transcription factor networks that were informed by multiple modalities. And when we look at those transcription factor networks, we see two transcription factors are changing between multiple sclerosis and the control border cells. Um, and these two transcription factors are key because they are driving these adherent junctional changes. So the border kind of the connections between cells, they're driving the changes in those genes. The third question I wanted to ask was, what's the mechanism that could be turning on these transcription factor like networks? So basically, I took things like proteins that had been found in the cerebrospinal fluid already in people with multiple sclerosis. So if we look at the cerebrospinal fluid of people with multiple sclerosis, what proteins can we find more there than in other cerebrospinal fluid? These proteins. And then basically we asked kind of which of these are predicted to be driving the adherent or junctional genes through experiments that have already been ran. And we built we build a matrix and we see that two specific proteins that have already been found in cerebrospinal fluid are driving the junctional gene changes, which are here in the border cells. And that's important because these proteins that have been found in the cerebrospinal fluid, when they bind to cells, they're known to activate these transcription factors, which are driving the adherent junction changes. So in summary, we found a mechanism that linked kind of that of how border cells could be changing. So these two ligands bind to cells, activate these transcription factor networks, causing their junctions to open. And basically, to visualize this, we took tissue from these patient brains and we looked at the border cells. So this is the border cell that I kept talking about. It's called an ependymal cell. This is the cerebrospinal fluid, and this is the rest of the brain. So this is what a junction should look like in a healthy person. But when we look at the same border cells in a person with multiple sclerosis, you can see this is the cerebrospinal fluid now. This is the brain. This is the junction that was once over here. It's completely split open. So it's a visualization of the junctions opening. This is us just staining for proteins and seeing that the proteins that make up the junction go away in multiple sclerosis brains. Here we did an experiment basically to show that basically what we did is we took people's brains We took mouse brains, we injected them with the protein interferon gamma that you see over here. Um, and when we inject this protein into the brain, you can see that a fluorescent dye that we put in the cerebrospinal fluid diffuses through. It kind of recapitulates the gradient that you see. Um, in a healthy brain, when we just inject the dye, you get a normal kind of, it stays in the cerebrospinal fluid. But then when we knocked out the interferon gamma receptor and injected interferon gamma, and the fluorescent kind of dye, everything stayed in the brain. So we found basically we proved the mechanism that we found in the wet lab. But I'm using this example. I kind of wanted to bring it up because it's an example from my life that I talk about, of how theory and understanding the genes that you're looking at can kind of guide discovery. And it's maybe not that exciting because that's how science had been done for like 200 years. But I bring it up because I think as you start using these kind of really modern machine learning techniques that you see over here, it's really important to say, like, why am I using this? And what can I get from this analysis? Um, uh, uh, because there's a million ways to tilt like an object of data. There's so many variables you can look at, and a machine learning agent or whatever can explore that space well. But just because of how much data there is, even in just a problem like this, you might not ever be able to tilt it in the right way. And you can use like agents can look at your literature reviews and it can, you can really help with that analysis. And I'm sure something like this might be automated soon with the right human feedback. But I just think it's an important example of how you have to be thinking about a problem. How can not only what are the best models can I use, but how can they inform the theory that I have? So yeah, now I'm
I
going to be
think
talking. Yeah.
I have a comment. I think you also brought up
Yeah.
like a deeper biological question. Sorry, a deeper biological
Mm,
um node, here as well. Especially with the transcription, factors. And
mhm.
this is something I did not realize until I got into the grad school, in the sense that transcription, factors work together, right?
Yeah.
It's it's like a network, right?
Mm,
And I think, previously I was thinking of it as a one-to-one, mapping. A transcription factor binds, some, DNA elements, recruits other transcription, machinery, and then
mm,
causes the expression of, the genes. But then, in grad school, I think what I found, was that you could actually, have, multiple transcription factors driving, the expression of a particular gene, or even
yeah.
multiple transcription factors driving multiple, gene expression, which brings up the topic of pleiotropy.
Yeah.
So I guess that's why, Finn was, talking about networks of, transcription, factors rather than, individual transcription, factors. Am I right in my, thinking?
Yeah, totally. No, yeah. I think like a transcription factor is just like an on switch in the cell. And like it's intuitive to think that one on switch does one thing. But the thing that makes biology so complex is like there's a million on switches for a million different things. And I think really my general rule in biology that I think about is anytime anyone ever tells me one thing is right, 99% of the time they're wrong. You know, I think I think the answer is always maybe, and the answer is always it's the mix of things.
Mm.
Sometimes one specific factor, one specific thing might be way more important, but the answer is always it's the mix of things, in my experience, with a couple very rare exceptions. But yeah, I think just always consider nuance because it's always there in biology is like the main thing.
So basically,
Um,
you can make conclusions, but like have room for maybe things go wrong or things might change.
Yeah, and I think that like randomness is what makes especially biology so exciting. I think it makes it more creative. It makes it more kind of makes more possibilities to build on things. And if it was just like a race to figure out if it was just like a race to build all the Lego blocks of like our world, I think that would be less exciting than how like kind of creative it is now. Because
Right.
you can't just build Lego blocks in science. It's more random and cyclical than that. You know what I mean? It's like a,
Right.
you know, anyways, yeah, I could go on and on about that, but yeah, thank you for bringing that up. Um,
Mm.
yeah, so now I'm going to be talking about an example of how machine interpretable black box data can accelerate science. So before I talked about kind of how looking at genes and kind of using data that you can understand can really help your science. So either like the analysis you're doing or what agents are doing, because you can inform your kind of investigation by biology and theory, and that can lead you to a conclusion really fast. But what's interesting in kind of how the field's progressing is there's actually a lot of data that's really informative that, humans might not actually be able to understand because it doesn't necessarily have like an interpretable component to it. Um, and I think it's a really important thing to consider for yourself and for the agency you use, um, because uh, you know, you have to think like, what's your goal? Like, why am I doing this research? And potentially, if you're using data, if you have enough of it, but you might not totally understand it, but if the fact that you have so much of it, it might actually help a lot of biology in a unique way. So I'm going to be talking about an example of how I'm using data that I don't fully understand, but a machine does, and it's helping my research. Um, so yeah, this is the work I do at the Arc Institute and the project I was selected to do. I'm building something called phylogenetically aware single cell spatial proteomics. That's a big word, but I'll walk you through it. So don't worry. But yeah, I would be scared about that big word too, or big couple words. But yeah, anyways, so there's a one-to-one ratio of human to bacteria cells in the body. So bacteria cells they're an essential part of the human biology, even though they're not human cells. And because not all bacteria are bad. Some are good, and some work with human cells in ways that we don't understand yet. But they're essential to the human body, that's for sure. So the single cell, like the thing I talked about before, like single cell, where you're looking at the transcripts in a cell. Um, that stuff's really cool. But it actually a lot of the time only lets you look at human cells. And that's an issue because of just how, like for a lot of reasons, but just because of how much bacteria you are. If you're telling me that something's going on in a specific tissue you're looking at, that's cool, but it's like you might be ignoring half the cells and like even if you're looking in the gut or something, like maybe there's like a 5-to-1 ratio of bacteria to human cells. So you might be even acknowledging like like 5/6 of the cells. You know what I mean? And I think this problem is interesting because um yeah, like what I'm working on is interesting because uh what we're trying to build with this kind of black box machine interpretable data is a way to look at human cells and bacteria cells at the same time, using a signal that I'm going to talk about that is not actually fully interpretable, but it can help us in a unique way. So basically, the reason that kind of the RNA transcripts, like looking at single cell RNA sequencing data, doesn't really allow you a lot of the time to identify bacteria is because bacteria have different types of RNA. So they have really short RNA. It doesn't have a polyadenylated tail, so AAA at the back, and it's often really low in abundance. So there's not a lot of it. And that makes it really hard to detect with single cell sequencing, this kind of standard workflows that exist. So kind of our group was thinking, how could we get around this? How could we find a way to identify bacteria within a tissue in a new way? And so what we're doing is basically we're using proteomics. So have you guys heard of maybe talked about mass spec before, Samuel? Mass spectrometry?
So not here, but like
Okay.
people
It's
in
discussed.
Ghana may have gone
Mass
through
spec.
mass spec, as an analytical chemistry technique.
Cool. Yeah. So I mean, I'll talk about it anyway, but I was just for my background, for my like understanding. So, basically, Yeah, so mass spec is a way to detect proteins in tissue. Um, and it uses kind of some physics and stuff like that, but fundamentally it's just a way to detect proteins. And what's interesting about proteins, I'm not sure if you guys know this, but everything that's living has proteins. Human cells have proteins, bacteria cells have proteins, and you can kind of detect them with the same, just everything has proteins. So what our group was thinking is, um, could we potentially look at proteins instead of RNA to tell if a single cell is from a bacteria or a human cell? Basically, we're using a technology called deep visual proteomics. So this was developed by one of our collaborators. It's pretty cool. Basically, you take a piece of tissue, you fix it. So you essentially glue, you can think of it like gluing all the cells in one spot and all the proteins in one spot. Then what you do is you stain all your cells with immunohistochemistry. So that's what you're seeing here. So you can tell what are the cells that I'm looking at. And then you can use AI to find a mask or like find the border of each cell in your tissue. So like here would be a border, here would be a border. And then the fourth step is you take a laser and you cut out each of the individual cells like that. So you cut it out and then you cut out that cell with a laser, and then you catch each individual cell for mass spectrometry. So this is a really cool modern technique that one of our collaborators developed, and it allows you to build um, single cell spatial maps of tissue. So basically, you remember where each cell you cut out was cut out, and so you have the XY coordinates, and you know where it is, which well you caught it in. And then when you send it to a mass spec sequencer, you get the protein data, and then you remember the XY coordinate, and then you can build a single cell spatial map of of cells with their proteins. So it's a really cool technology. And like I said, we wanted to we were thinking, could we use this signal to tell again if a single cell is from a bacteria or a human cell? And yeah, basically this is just an explanation of mass spec. So basically you take a cell that you caught in one of these wells that you cut out with a laser, and you run it through a liquid chromatography column. So this is a way of separating different proteins to get like kind of similar groups. So analogy I would give is like, imagine you put water on sand. The water would go really pretty fast through some of the sand, and then you'd get water at the bottom. But imagine you put like honey on sand, for example. The honey might take like a million years to get to the bottom of the sand, but it might get there eventually. So basically, what chromatography does is it takes little beads and it puts them in like in this column, and then you put your sample that you caught, like your cell that you grounded up, put it in here, and it separates the different proteins, just like the sand would separate water from honey, you know? And so you get groups of proteins. Then basically what you do is you take one of these groups and you send it to the mass spec detector. And it essentially, what the mass spec detector does is it looks and says, how fast does a protein go through like a magnetic field? And basically, proteins have like charges, and basically the charges determine a lot of the time the relationship between the mass of the protein and the charge of the protein determines how fast it can go through the detector because of the magnetic field. And so you can get a readout of each protein in this column's mass and charge, and you can also get a readout of how much of that you detected. Because there could be multiple proteins in one of these columns. So the intensity is how much of that one thing you detected. And then basically what you do to get to squeeze out a little bit more information to help tell you what the protein you're looking at is, you take one of these peaks and you break it up. You blast it with ions and you fragment it essentially, like cut it up with scissors. And then you run it through the same mass spectrometry detector as before and get a mass charge ratio for the fragments of the protein. So like the parts of this protein that you cut up with scissors, you get a kind of a readout of the fragments, and it's the same thing. You have the mass charge and the amount of the different fragments you detected. And standard, like the standard approach is you use like algorithms to align these kind of mass charge values to a reference genome to tell you what the protein you're looking at is. So you basically you have a reference and you say kind of what looks like what I've seen before. And so you align things. But what's really curious about, and what our group thought was like kind of a missing in this is that what's curious is a lot of the time about 50% of the peaks you see here are just thrown out in the analysis because you can't align it to a protein, to a human interpretable feature. And that's a huge missed opportunity in our view because you're throwing out like 50% of the data. We think that's pretty crazy. And what our group said is in order to detect if a single cell comes from a bacteria or a human cell, we could potentially train a deep learning model um, if we give it the right objective to tell us if a specific cell we cut out is from a bacteria or a human cell. And we do this by giving the right objective and letting the model reason over the raw data input space and not over the human understandable kind of input space and the features. And so that's an example of what we're doing, and hopefully that clarifies the title a little bit, the
Yeah,
long title.
this is really cool.
Yeah.
Yeah, if I may interject also to
Yeah,
give
totally.
a little bit of clarity. You have a tissue, maybe let's say skin tissue.
In tissue, yeah.
Right. Ethically, you don't do this to yourself. So you have a skin tissue, and then put it under like a microscope that's able to do, I mean, use, lasers to sort of like first, scan the cells and see the spatial locations of the cells, and then sort of like cut it out, cut out individual cells into specific, wells,
Mhm.
and then you try to, for each cell, probably break it open and measure the proteins, of, each cell. I guess the question which I would ask, which I'm sure other people, may also want to ask, is that why is the spatial part important, in this, type of way? Yeah, I mean, why didn't just do the, chromatography to, get a spatial cell proteomics? Like, why is the spatial part an important, aspect of the work?
Why is the proteomics part
No,
specifically
no,
important?
the, spatial, part, because
Oh,
you want to
spatial
know the
part.
location of the cells, as well. And I'm
Yeah.
sure people might be
Yes.
thinking about, like, why is this step even important or, like, relevant to the objectives? Yeah.
Yeah, totally. I mean, honestly, I have three reasons.
Mhm.
One, it's really cool. Two, um, the real kind of one of the real answers is that cells don't exist in the ether or like just in space. They exist in tissue. So like the relationships between cells are really important. And a lot of the time when you're doing single cell, you lose that relationship. And what's really cool here is not only are we looking at human and bacteria cells at the same time, but we're able to look at how they interact and where they interact. So it's really informative information. Um, and then the third reason is that this is one of the only ways to get single cell proteomic data. There are other ways, but what's really cool about this is, we're able to make sure that we're getting single cells, and even cooler is we're actually able to make sure we're getting bacteria cells because you can stain for bacterial markers, like bacterial ribosomal RNA with some sort of fish, whatever. You can stain for bacterial markers and make sure you cut them out. So you're not like only getting human cells or you're not losing bacteria cells because of the way you're doing it. Yeah, so three answers. It's cool, you preserve spatial information, and you make sure you're getting bacteria cells.
Great.
Um,
Thank you.
yeah.
Yeah.
Sweet. Yeah, but so again, this is an example of how you can give data to a model that you don't actually understand yourself because it's in this weird, like, what the hell? I mean, I don't like, I'm still learning, but I don't even know what the hell this is. Like, you know what I mean? How you can give kind of crazy looking, uninterpretable data to a model, and you can learn something even if you don't understand what it actually is. Um, and it's important, I think, as you consider what problems to give to do yourself and give to your agents with this amazing cloud science initiative that you guys are talking about. And so this is just a view, just like very high level of what our model looks like. And maybe I still don't even understand everything we're doing. I'm trying to figure it out. But hopefully you guys can just catch a vibe and learn maybe some new terminology that can help you, even if you don't understand the whole model, which I don't either. So um, basically, so for a given cell that you cut out over here, you have the mass spec data that I was talking about. And then you have a vector of the first time you put it through the mass spectrometry data, what's the mass charge and the intensity. So this is the mass charge, like the x-axis, and this is the y-axis, like just like over here. Mass charge, it's actually over here, like the mass charge and the intensity. So you have your mass charge and your intensity. And then over here,
Protein, right? So
yeah,
yeah, of
exactly.
the
For a
protein.
given protein, for
Yeah.
one protein.
So
Um,
just to
yeah,
clarify, this is the cell that Finn is showing.
yeah.
You're gonna fragment the cell, get the proteins, and you get like something beautiful on the right hand side. But then I think the explanation he's also giving is that this S is basically the protein charges that is coming
Yeah,
from a single cell,
it's
right?
like just like in a single cell where you have a cell and the amount of genes, like the amount of gene one it has, like it
Right.
maybe has 10 counts of gene one. This is one cell
Hmm.
and the amount of protein one it has.
Great,
So this is the math way of looking at the protein.
great,
Um,
great. Thanks for clarifying.
Yeah, no worries. Thank you for asking. I really care about this stuff. So I'd be really mad if you guys didn't come away with something. So please stop me at any time, and you'll notice that I'm really redundant in the way I talk. So I think but I think redundancy is so important, and just please ask anything that I haven't clarified because I think it's cool. And I'd love if you guys took away something. So yeah. Yeah, so this is one protein that we got from this cell. And this is just the way we look at the data. So you represent the initial thing, and then it's just think about it as one protein. Then what we do is we put it through something called an encoder, which is like a way to turn this representation of one protein into something that's informative. Because this is just a bunch of numbers, but like, you know, maybe we could turn it into a more informative representation that actually tells the machine learning model what it is, in a more abstract way than just numbers. So an example I'd give is like, okay, so um, you know, there's a lot of data that exists in the world, right? Um, but some of it actually carries a similar signal. So if I wanted to represent kind of um, if I wanted to represent kind of my height, my shoe size, the length of my dad's favorite movie, and like how long it takes me to walk to school, you don't actually have enough axes for that, right? There's four variables, and so you can't represent it in 3D. But something that a lot of people do in machine learning is they say, wait a second, my shoe size and my height are kind of correlated. So maybe I could just collapse that to one axis, and then I could represent it in 3D. I could have the length of my dad's favorite movie, the time I take to walk to school, and then a variable showing the combination of my height and my shoe size, because they carry the same signal. So basically, what this encoder is doing here is similar to that. It's saying, how can we represent a lot of different proteins, so not just this one S, but a lot of different proteins on different axes that represent kind of common signal. So like maybe one axis could represent the size of the protein. One axis could represent um, whether or not it's a kinase that does phosphorylation, for example. So we're doing an encoding that just gives us a little bit more information than just the raw math. Just like the example I gave. Then basically what we do is we train basically after taking all the proteins and encoding them, so putting them on like an informative axis, we train a predictor to say on the like on the it's called tokenized data. Um, like on the informative axis of each individual protein, we train something to tell us is a given set of proteins, like a bag of them, are they like, for example, from this one cell, is it from a bacteria X, bacteria Y, a human cell? So again, we take a cell, we take its proteins, we put them on an informative axis, and then we take the groups of proteins from this one cell and say, where is it from? What is it from? Um, and then basically this is how we train it, but we can talk about that after if we have more time because I'm conscious we're coming up on the time limit. Um, but yeah, basically, this is an example again of how you can give data that you don't actually fully understand yourself, and take advantage of the fact that there's a lot more of it than data you do understand and learn something from the data. And just one more time, as you're thinking about what problems to give your agents and to consider your cells, this is another thing to consider, where you could learn really cool things. And yeah, I think it's important. So I'm conscious we're at 45 minutes now. So I wanted to give you guys the option of choosing potentially like a last topic that I could talk about. It's kind of a choose your own presentation to make it fun. So I do a lot of philosophy of science, and I kind of have been thinking a lot recently about my philosophies on agentic science. And so I'm happy to talk about that to you guys. But also, I recently started kind of researching with like some of my friends at a startup that they co-founded, and it's based off the analysis that I already talked about. So I was hoping that I could kind of use this white box, black box data paradigm to talk about one of these subjects. So I was curious what you guys wanted to hear more about. Can we do like a vote maybe in the chat or something, or we'll see if it works. If not, we'll just take whoever responds. Startup, startup. Okay. Maybe we'll give like two. Okay, it sounds like everyone wants to hear about the startup. Both. Well, okay. Well, well, let's see. I'll start with the startup stuff, and then if we have time, we can talk about the philosophy of agentic science that I think about. Um, so basically, uh,
I think
so
you
what we
already
do,
knew people were going to go for the startup.
startup. Okay, yeah, you makes sense. So, um, basically, so this is, it's a startup we recently found. Like I didn't co-found, but I'm like a lead researcher at. And basically, the what we're doing is ML, like machine learning, drug discovery, and clinical trial redesign. So again, just some words, but hopefully it'll all make sense by the end of it. But basically. Yeah, and I have to mention we got funding from Y Combinator, Glasswing Ventures, and Pear VC in my presentation. I have to say that. So
Congrats.
also disclosure, I have conflict of interest, so maybe don't believe everything I'm saying. It's up to you guys. Use your own best judgment. Um, but basically, yeah, so um, a clinical trial is a way of telling whether or not a drug works or not for a patient population. Basically, what you do is you take a preclinical sample or like some sort of biopsy, and you say, based off this biopsy, based off this marker, based off these symptoms, I'm going to enroll this patient in this clinical trial and see if a drug works on them. So you give like some of the people in the clinical trial a drug and some people a placebo. And then what you want to see is does the people that were given the drug, do they respond above the placebo group? But the issue is that this is like a really kind of it's a really simple test. It's just does one group, it's like a null hypothesis, like a p-value. Does one group respond above another? But what happens all the time is that in the people you gave the drug up here, some people respond and some people don't. So you actually lose kind of a lot of the signal and it kind of gets brought down to the same level a lot of the time because some people respond to the drug and some people don't. But what we said is actually you remember the preclinical sample that you used to enroll people? you basically let that go after people are in the trial. But what we say is let's actually try and use that to predict which of the people that got the drug respond and don't respond. And that's a kind of a thought experiment about why that's useful is like imagine a case where you had a drug where 10% of people it worked for. But what if I told you that based off the preclinical data, I could tell you 95% of the time who the people that respond to the drug and who the people that didn't respond to the drug are. So even though it doesn't work for a lot of people, I can tell you who it works for. In the classic clinical trial design, that drug would not be on the market. But I feel like what we argue is that's actually a little silly. If we can tell you who this drug's going to work for, we should use that drug. And that's essentially what we're trying to do. We're trying to build machine learning models that take preclinical data that's usually just thrown away and both discover drugs that should have worked in the past and help discover new drugs and redefine kind of the clinical trial pipeline. Um, and so we can basically, it's kind of this phase two drug to phase three drug trial transition. Trying to see if we can make both rediscover drugs and find new ones. And so in one of the ways that this is useful and what this graph here I'm showing you is basically or like what this graph here is showing you is what I'm talking about. So basically here, this pi value shows the amount of people that were given the drug that actually respond. So here, 10% of people given the drug respond. Here, 20% of people given the drug respond. Here, 30% of the people given the drug respond. Here, the x-axis is how well we can predict who responds to the drug. And in the best case scenario, for example, like for us at least, for making money, um, like uh, if we build like a model that can use preclinical data and predict with 90% accuracy who responds and who doesn't respond to the drug, when the drug works in 10% of people, basically, if you map that over here, this shows that we could save a clinical trial about 90% of their patients that they enrolled. We could shrink the clinical trial by 90%. And so we showed in a recent blog that we wrote, which I'm happy to share with you guys, that in a specific example, we could have saved $15 million and 500 participants enrolled uh, for um, for a clinical trial that was done in the past. Um, and I think it's an idea I'm really passionate about because it can save a lot of people's like from being enrolled in risky things. It can save companies money and it can discover new drugs to help people in the healthcare system. Um, and I think another thing that's really important is that a lot of clinical trials are done in the Western world on like white people pretty much and where these hospitals have resources. And there's a lot of kind of communities and health communities that don't have the resources to do clinical trials. And another kind of use for this is working with people that want to do clinical trials but have this major barrier of recruiting cohorts. So we're trying to work with a lot of people in India right now, like a lot of hospitals in India right now to help them with some of their clinical trial design and really shrink the amount of people you need to enroll. But I think the conversation could also extend to East Africa and like different kind of clinical communities that are interested that maybe have a little bit less resources than people in like San Francisco, you know? Um, uh, and yeah, so it's interesting stuff. But one of the things I think about a lot in this kind of world is how do you make yourself a product rather than a feature? So this idea is great, right? But theoretically, everyone that has a machine learning research unit in their drug discovery company should be doing this. I don't think it's being done because there hasn't been this translation between foundation models and like clinical trial design yet. But once the idea gets out, we kind of expect that it's going to spread pretty fast. Um, so it's a difficult spot for us. So I think something we think a lot about is how do we make ourselves a product rather than just a feature that someone else can do. And actually, what kind of the current direction we're going in is based off the research that I showed you from the Arc Institute, the black box data thing, where we're essentially taking clinical modalities where most of the data is thrown out because it's not interpretable. It's not like human understandable. And we're using the data that's thrown out to build really informative predictors. So an example that we're doing right now is based off proteomic data. There's a specific drug trial where they tried out a drug on people after collecting biopsies from tumors and then doing proteomics on the tumor biopsies. Um, what we're trying to do, but again, 50% of the data from that tumor biopsy is thrown out. It's crazy. Um, and so what we're trying to do is use the same paradigm I'm talking about with the encoding stuff and building informative kind of axes for the data. And we're trying to develop a model to predict who responds and who doesn't respond to the patient. And it was a phase 2 trial. And we actually think that there's like we're getting some preliminary results that this trial actually could move to phase 3 if we have an informative predictor, and we could do it in a lot safer of a way. And so, yeah, that's basically the startup stuff that I'm doing that's based off this paradigm of it, black box data that I talked about. We have 5 more minutes, so I'm maybe do you think we have time to talk about the other stuff or no? Do you want to have questions? Also, I'm happy to stay longer if people have more questions, but what do you think, Samuel? You're the chair, I guess. Oh,
I think, can you hear me? Hi, great work. I'm trying to think, Sam and the team, how the ideas that Finn presented today might be applicable to the research in Ghana. Is there some stuff that I'm just open the discussion. Um, do you have kind of ideas or data sets that are big enough to be um, that can be used to reveal kind of some kind of can be tokenized, can get some insights from similar methods that Finn presented. So, and Finn, maybe you can also say what general data can be used. I know that you were talking about mass spec and so on, but for the encoder decoder,
yeah.
what what is actually needed?
Yeah, I mean, I think theoretically anything could be used. Anything where you have variables and you're trying to predict what how whether someone responds to a drug or not can be used. So H&E data, like histology data you can use genetic data, you can use bulk single cell data, you can use cell-free DNA, like data, clinical data, it's just data. You know what I mean? And if you can build a good predictor based off it, that's the only prerequisite, in my opinion. So you can work in a lot of situations, this general idea. What we're trying to do is basically, the word is like alignment-free black box data prediction of who responds to a drug and who doesn't. And what we're doing is we're trying to find modalities that have historically kind of been ignored because they haven't been kind of human understandable. Actually, something I should mention just while it comes to mind is that you can actually go back and find what the features the model used are after, and figure out what signal it was learning from. So you can still learn informative biology, but what we're doing is we're working a lot with cell-free DNA, um, bulk proteomic data, and we're actually looking to start working with bulk single-cell sequencing data. Something that not I think a lot of people realize about single cell data is that it's kind of actually the same situation a lot of the time where 50% of the data is thrown out. Basically, the raw input files for single cell are something called a FASTQ file, where you have a lot of reads. But then you essentially take that data and try and align it to genes like coding regions, like open reading frames. But a lot of the data you find actually can't be aligned. And so you end up losing a lot. So another thing we're trying to do is build models that can reason over raw FASTQ files by creating informative embedding. But yeah, for Samuel and for people in Ghana that like maybe want to get involved or like feel free to reach out because we're looking for collaborations and to work with really nice and exciting people like I'm sure you all are. I think it's really anything that exists, and anything where you can have where you can just have data and you want to predict who responds and who doesn't. Yeah.
And then if you go back to your startup, can you go back to your startup work? what is actually the model that you use here? Is it the same kind of structure architecture as you presented before, or you modify it for the problem? And I
This
guess it's not a foundation model because usually so it sounds like you are really dependent on the levels or the predictors, or it's a foundation model that you then fine-tune it for the task. So
Yeah, so I think we actually use a lot because, we do use foundation models for doing this prediction to make informative embeddings. And that's like it's a great use of foundation models. But these days, I actually don't believe, I don't believe anything I see unless I've seen a linear baseline first. So someone that's trained a logistic regression on the raw input space to see how good it is. So because it's really important a lot of the time in these really like high noise kind of low data regimes to see like a lot of the time the best prediction you can get is a simple one because you overfit to the noise is the word. But basically, to answer your question, we use everything to show to both give ourselves a baseline and show kind of the utility of a more modern foundation model approach. But we have our own bespoke foundation models we made for RNA data with like the virtual cell challenge at ARC, but yeah, we're doing it all basically. Because every step is informative, like every great scale up of model.
cool. And I'm still trying to understand how so maybe I missed a slide or didn't pay attention. How
Yeah, no.
the deep learning entering here? Like where is the model?
Yeah, so the basically this is like a figure we took from our recent log where we were doing single cell data. And we built like a diffusion model based off kind of public single cell data and like the Tahoe therapeutic data sets to create embeddings, basically, create raw embeddings of gene data. But like another example would be here. We're using kind of this really similar architecture that I'm building for my work at the ARC. We're using the same architecture to create informative embeddings of proteomic kind of data, like liquid chromatography, MSMS, proteomic data. So deep learning comes in every time you want to make out an informative embedding. But the actual prediction, a lot of the time, we're using really simple models, but also more complex ones to see what we can squeeze out.
Cool. And okay. And any questions from the crowd?
Oh, Samuel, I think you're muted.
There was a question from Mehdi. Can we say this behavior is because of quantum mechanics in the cell? And I think that's a question about
Yeah,
explanation of the pink cell where you were explaining the model for the
yeah,
spatial
yeah.
transcriptome.
So I think that, you know, everything like the way you look at a cell, you can look at it in many different ways.
Mhm.
And one of the ways to look at it is using this kind of protein data that, yeah, has a lot of like kind of really small, like really small features that are involved. So like little kind of fluctuations in what the protein looks like at a really small chemical level. So kind of like quantum. And yeah, part of the signal we're getting is because we're able to learn stuff about the really small changes in the proteins, like the kind of quantum changes in the proteins, that can tell us something. So yeah, to answer the question, it's a great question. Yeah, a lot of what we're able to do with this startup is because we're able to learn about the really small changes in proteins.
Great. There is another question. Can we join your team? We are open source research guys. Our domain is computational biology, currently working in bulk RNA, and we'll love to see if there are openings. So this is from Tanish. I hope
Yeah,
I'm
totally. I'll send our website.
yeah,
But I was gonna say, we're looking for a collaboration from like all over the world. Anyone that can like kind of give insight into kind of how data is collected in their own community,
right.
give resources about like kind of where our work could be helpful. we're in the process of learning right now. So we're not exactly sure what we're gonna do, if we're being honest, but we'd love to hear like from people that think they could kind of help us learn about what they do. And yeah, I think everything's informative. Why don't I actually give you guys my Samuel, can you give them my email to just
Yeah,
reach out?
would I would
Sounds
I would drop it, but I'll drop it in the chat. All right.
sounds good.
So that'll be good. Just trying to make sure that we officially close, and anyone who wants to stay on for more questions, to Finn, you could stay on for more questions for Finn, but
Yeah, please.
because I'm sure other people may want to hear your exploits in the philosophy of science also.
Yeah, totally.
All right, so this is Finn's email, and yeah. So I think with that, like we've officially, closed, today's session. Thank you so much, Finn, for like really taking the time to really, talk to us about your work, and and also like about, but not just the science, also about commercialization of the science, as well, in terms of
Yeah, totally.
study. I think this is great.
Yeah,
Um,
totally.
yeah, and I'm sure like would want to hear the other parts of what you are doing. Just a shout out. There's a website. We have a new website, now. You could,
Awesome.
really check it out, and give us, feedback on it. All right. So
Yeah, totally.
yeah, if you scroll down, you see yourself if you scroll down. So
Okay. Oh wow. Oh my God.
Oh, yeah. Oh, amazing.
Wow, it's so cool. Was this how much did you do this with Claude Science as well?
Yeah,
Or?
no, this is just cloud, and I use like some part of it was with Grok 4.5. It's super fast,
Oh, cool.
and yeah, I mean, if you go, there are basically opportunities, as well. Like if you're looking for hackathons, if you're looking for, things you want to apply for, there are some, few things here. I want to bring like, something called open, problems, or like, really tough, problems that would just, be open, and like, people who want to take on like, very great challenges with great reward could also,
Yeah.
look at it, and we could
It-
connect you to maybe other, stakeholders as well. Yeah.
yeah, totally. I mean, I have like a lot of you know, actually this reminds me a lot of someone we met at CSHL's website. I think he would be really informative. He's doing his PhD at Oxford, so I'll send you
Okay.
his LinkedIn after and maybe send an introduction, but I think he has like he's built a website of like open problems in science that he's like mined with agents, and I think it'd be
Right.
cool maybe to like integrate in
Yeah.
here. I don't know.
Yeah.
Just
Yeah. I think that'd be.
but yeah, it's really cool. So yeah, I'd love to stay in touch on this. And if do you guys have like a channel or something where you send news or
So I think for global channel, so we have like a small WhatsApp group, but I think, for
okay,
the
cool.
global, channel, we may have to do something like maybe Discord, something. So that's still in the
Okay.
works, and I'll let you know once it's set up.
Yeah, let me know. I'd love to be part of any community
Yeah,
like you.
yeah.
So
Because we would want to have people from India, Africa, US,
oh yeah,
Canada,
nice.
China, all of that. So I think, that would be great. All right. So this concludes the official session of today's talk. If you want to stay for the vibes, you are welcome. I'm going to stop the recording now.